rheumatologist at the University of Bristol. And this year in Florida, so in 2026, I presented at this presentation at the ORSI Congress, and David Hunter has asked me to record this again for the Osteoarthritis and Cartilage Journal. So this talks on published examples of dubious osteoarthritis papers and how we might stop them. I have no financial relationships or commercial interests that are relevant to this, and I don't discuss the use of any off-label drugs. So when putting together this paper, I thought to myself, what is a dubious paper? This is a very subjective decision. To some, what to some might be very dubious, to others is maybe just low quality research. And so when I thought about looking at the subject, I decided that there was a scale of dubiousness. And so this ranges from the not very good research at one end of the spectrum through to sort of really fraudulent and fictional research at the other end of the spectrum. And I think it's important to recognize that this isn't a black and white issue, that some research does more harm than others. So along with this scale of dubiousness, you've got a scale of harm. And I think there's lots of not very good research that does very little harm. That's not to say it's no harm, but it's less harm. But there's some real examples of very severe fraudulent research that can be very harmful. And I think it's mindful when we review the literature to think of both of these aspects as important. So then, how do we decide if a paper is dubious or not? And I basically come up with 4 points that I look at when I'm looking at research. So, is the methodology correct? Have they applied the right thing to the right data? Are they generating new knowledge? Or are they just rehashing what we already know for the 1000th time? How hard is it to do the research? I think to really push knowledge and science forward, it's likely that this is going to be very difficult to do. And so if somebody can do the analysis within an afternoon or a week, it's unlikely to be high quality research that we should be paying a lot of attention to. Or if it is good quality, we should probably be thinking that we require more of it, as in there needs to be more analyses to go with it to justify publication. And then again, does it do any harm? So what does it look like out there at this time? Well, I like to think, and I don't necessarily have a lot of evidence to support this, that the vast majority of dubiousness or dubious papers would be at the not very good end of the spectrum. These are still scientific analyses that have been done. They're just of low quality compared to the outright making up data, making up findings, writing papers, fraudulent end of the spectrum. I think that's a small percentage. But the presentation that will go alongside this from my colleague, Dr. Cindy Boer in Erasmus in the Netherlands, maybe one starts to think that the fraudulent end of the spectrum might be a greater number than I initially thought. If we look at a recent paper that was published in the BMJ that David suggested that I look at, here the researchers trained a machine learning algorithm on retracted papers, and then they put out this algorithm to detect these papers into the oncology literature. And what they found was quite incredible. 10% of papers in the leading journals were flagged as potentially fraudulent or made by paper mills, and greater than a third of papers originating from China were flagged by this. And I think it should be recognized that there's a lot of good research coming out of China, and the investment they've had in their science sector is incredible. But clearly, when you look across the research at paper mills, there does seem to be a problem with some of the research coming out of China. And is it simply that they are such a big country with a large population that they will always overrepresent, or well, they will always, or is it that the incentives and the structures lend themselves more to problematic research? I think more debate and discussion and research is needed in this area. So there was a blog that I found that looked at what was going to happen with AI and the flood of submissions coming with that. And if we think that the number of research articles published a year tends to be quite fixed, and if you look at the bottom here, the human submissions are the yellow bars, that's the number of submissions there are each year currently. If AI can through an order of magnitude lead to a huge number more submissions, it looks likely that what we'll see is a greater flooding of the lower end of the market here, and this could impact what research we see being published. Whatever sort of protection mechanisms you put in place, it's very hard just when you're fighting this number of submissions coming in that they won't get through. But hopefully this higher quality end of the spectrum will remain largely human written, but only time will tell. So I've given you a bit of background now about the state of play and what we think of as a dubious paper. So now let's consider some in the literature. And I think it's worth being critical at oneself when doing a talk. And if I'm going to potentially say that other people are publishing dubious science, I need to look at my own science. So this is a Mendelian randomization paper that I was second author on that was published in Osteoarthritis and Cartilage a couple of years ago. If I review this paper with my new scale in mind, did we use the methodology correctly? Absolutely. This is a really robust Mendelian randomization study done by Maria Largely, who was a very senior and well-qualified postdoc in Bristol. And we rigorously assessed the literature regarding the genetics of bone mineral density, osteoarthritis, and spinal stenosis to undertake these Mendelian randomization experiments, which potentially tell us about the causal relationship between these traits. Now, did we create new knowledge? I think we did our best to try and shine a light on gaps presented by observational literature, but ultimately we concluded that lots of these traits are bidirectionally related. So there was nothing groundbreaking in our conclusions. How easy is this kind of analysis to do is the reason why I put it up here. It's very easy, if you know what you're doing, to do these analyses within a week. If you have the coding pipelines and the open accessible data, which is now the case because genetic consortia share things across. So genetics consortia, when they do these genome-wide association studies, it's routine practice to share your data. And did we harm anyone with this paper? I don't think we did. So I think this is a low harm paper. But ultimately, do I see long-term this being a paper in its own right? No, I don't, because it's just not enough work to be put into a paper. What we need to be doing is using Mendelian randomization to triangulate with other analyses to really build a better picture of how traits are related. And why is this a problem? So if we look at the number of Mendelian randomization trials related to osteoarthritis published, you can see an exponential increase since 2020. And this isn't unique for osteoarthritis. We see the same thing if we look at total Mendelian randomization studies, which are now here on the y-axis. So there's this exponential growth, and what this is largely because of is openly accessible code and data. So you can download the packages required to do these analyses in R very straightforwardly. If you have AI or such as ChatGPT or Claude helping you with the coding, this makes it even easier. People deposit their summary statistics, as I've already said, online, so you can plug them in and you can do these analyses in an afternoon and write up a paper within a week. So I don't think that we want to have the world, every journal full of Mendelian randomization papers, and therefore I think Mendelian randomization is a good analysis tool, but it needs to be primarily done alongside new genetic data. So if new GWAS data have been published, do Mendelian randomization with it, use it in conjunction with other methods, observational studies, to triangulate data regarding to drug targets or epidemiological risk factors of disease. So if we move up our scale of dubiousness, we can now see, here's a Mendelian randomization study looking at genetically predicted T intake. So the key with Mendelian randomization is you have to have very good proxies, genetic proxies, of your exposure of interest for the methodology to be sound. And can you really adequately genetically proxy T intake, or is that very much societally and environmentally driven? I would argue it's the latter, and therefore I don't feel that this paper, for example, is a very good example of the methodology being used correctly. Have we got new knowledge from it? Not really. Was this easy to do? Very. Has it done much harm? No, because I don't think many people care about T intake regarding to osteoarthritis. But it's yet another paper that enters PubMed and clogs up people's systematic reviews. But I've talked about Mendelian randomization because that's the area that I work in, but we can think about lots of different examples of this. So we have the global burden of disease study, where we can see you can cut up this data for every different country in the world and publish a paper, it seems. And really, this doesn't add much new knowledge. The standalone Lancet publication every few years has the data that you need in it. So if you want to look up osteoarthritis within the United Kingdom, just go and find the supplementary data and put it in your grant application. You don't need to write a paper. We've got the N. Haynes study, again, huge number of associations here published, and then we have network meta-analyses and systematic reviews for any kind of small thing with regards to osteoarthritis. So this is looking at the effectiveness of physical therapy in osteoarthritis. So all of these studies, I'd say, the methodology may or may not be done correctly, but most of the time it probably is done correctly. It's just very easy to do with openly available data, and it doesn't really add much to the field. So at that lower end of harm. If we now go into the fraudulent end, this is much harder to prove. So the scale of dubiousness here is fictional and is fraudulent, and I think with this the harm goes up. So here's a study where they looked at stem cell injections into the knee. This got a lot of press at the time, and unfortunately the methodology they used wasn't correct. They didn't adequately test these stem cells on humans before they started doing these studies. Within osteoarthritis, little harm was done, but unfortunately these stem cells were injected into people's retina, which caused blindness. And then it came out that the adequate testing hadn't been done, and this paper was retracted. I think this paper got a significant amount of press before it was retracted. So people could have started thinking about doing research or even treating individuals with these stem cells when that really wouldn't have been the right thing to do. If we want to know about where we can find examples of these papers, Retraction Watch is a website that has them on. Here's an example of a paper that was presented at ORSI originally and then published in Annals of Rheumatic Diseases, a very high impact rheumatology journal. And what they'd done was they'd completely fabricated the number of animals involved in their study. which meant they were making up their findings. This isn't easy to do, though, because they still have to have labs with these animals in them, but the harm can be very high. So they claim that there were 12 knockout mice when there were not. And so the figure that I show you here was completely is a work of fiction. And later this, along with other work from the senior author, was retracted. If we think about other fields of harm, there's a very famous Alzheimer's study that has recently been retracted. But it was only discovered 16 years after it was first published. And it was only after a drug company whose drug, which targeted beta amyloid, was being sued. And in part of that litigation, they went back to the original study, and what they found was that the images had been falsified, and subsequently they found 70 papers with similar falsified images. So this is where you can cut up like Western blots and things to make it appear that you have expression when you don't. So this really was hidden in plain sight, but the field didn't pick up on it. If you've watched the very good Netflix documentary about this, you'll know the very sad case of the tracheobronchial transplantation, which was basically a synthetic trachea transplanted into individuals, and all the individuals died, but none of the individuals knew about the other people dying. So very high impact Lancet paper, show here with multiple co-authors, subsequently retracted, and a huge amount of harm done. So if we go back to this sort of scale of dubious, not good, and fiction, do we have a problem between wet and dry lab research? And I think we might do, because essentially with dry lab, you've got the ability to do analyses that with openly accessible data very easily. So that's your Mendelian randomization, your global burden of disease studies. So if you need papers, you can seemingly do that quite simply without actually making up data. Whereas I think with wet lab, it's much harder because you're running a laboratory. You've got all the costs associated with that. It's very unlikely that you completely make up the fact that you have any animals that you're studying. And I think it might also come from the fact that wet lab research takes longer. Scientists get less output. They might be under more pressure to make something work if the last 2 years of experiments hasn't been fruitful in terms of positive findings, and more pressure. But so this is just my own opinion to throw it out there, but I think it is worth considering when we're looking at these dubious studies. And then what can we do about stopping them? Well, I think at the not good end of the spectrum, we've got to look at ourselves as academics in the field and think about how we're reviewing papers. Can we desk reject papers? Can we very quickly spot problems that we can flag to editorial teams? So for me, at my example, it might be Mendelian randomization study that is done with openly available data that I just don't think should be a paper on its own right. And then you can just quickly say, this isn't enough for me to justify it being a paper. Don't even send it out for peer review. Is it adding knowledge? And I think that's the key thing. Do we think the paper's adding value? But even then, this does take time, and I acknowledge that. If we require payment to submit a manuscript, would this improve the quality? I think it will decrease the number of submissions, but if you're willing to pay a paper mill to write a paper for you, you're probably willing to pay a small submission charge. So I'm not sure this alone would solve the problem. And then obviously the crux of it is if you require papers to get promoted and the promotions bring more money, whether that and if you're an institution, you get more grant funding or more governmental funding if you have more impact and impacts judged through papers. You are just shifting the game towards publishing, and therefore you're really incentivizing these poor behaviors. Obviously, the journals are incentivized to publish papers because they're paid open access fees for many papers that they publish, and if not, they're paid subscription fees for people to see the huge amount of public papers they have. So I don't think necessarily looking to journals to solve this problem is going to be the answer. In terms of fraudulent papers, I think there's various things that you can do, like looking at the images, making sure they're not faked. I know journals now have software that they can do this with. I think you can. We probably need to think within institutions to make sure there's peer review and audit of people's papers and data, and making sure that there is that the data behind claims can be justified. And maybe again, you've got to look at the structures within an institution and incentives. If you're if you're pressurizing people to publish, then you're going to potentially lead people to making poor decisions and making up data just as they see that as the only way to keep their job. And maybe we need to look at better, more holistic ways of measuring academic performance more holistically, so that people don't feel under such pressure. So just to wrap up this talk, I think we've identified the problem. I think now it's the case of gathering stakeholders together to tackle the problem. We've got to keep monitoring it. And ultimately, I think if we really want to solve this problem, we're going to need to fund policing of academic literature and findings. So hopefully you enjoyed this talk. This was just food for thought. And if anybody wants to contact me about it, please feel free to email me, ben.faber@bristol.ac.uk. Thanks.